done the prokaryotes in as much as we're going to do them too. So I'll show you, we're focused now on just this part of Opisthocons, this presumed sister group to fungi, right? And this includes animals and a little group here I'm going to mention really briefly because it's going to come up with one of the questions called choanoflagellates, which are almost like little individual sponge cells. For a very long time people thought they were closely related to animals because they basically look like individual cells that make up a sponge, right? So there's like a unicellular version of a sponge. Yeah? So are you excited? We're almost all the way to the end. Okay, so I know most of you probably think I don't like animals very much. Do you think that? You think, oh, he just likes his mushrooms. He doesn't like animals. What's wrong with this guy? But animals really are arguably the most spectacular radiation of any living group. They are truly stunning. They figured out how to fly. They occupy every different kind of aquatic and terrestrial niche you could possibly imagine. More than probably 70% of the species that are described are animals. Most of those are insects. Most of those are beetles. Everything from a human to this fish to these little corals. Everything from a hummingbird to a blue whale. And there you're just talking within one phylum of animals, right? Just tremendous diversification. They take anatomical complexity to an extreme, right? There's no fungus, there's no plant that has the type of anatomical complexity that, and even you could argue some of the simplest animals. You realize that there are animals like rotifers and gastroctrix that are microscopic, that are smaller than those unicellular paramecia and amoebas that you looked at. There actually are animals that are smaller than that. And then, of course, arguably the largest single individual organism on Earth is something like a blue whale or something. You could argue that a fungal mycelium might exceed the size of a blue whale, but then you start to run into problems in determining whether or not you've got one individual or multiple individuals. At least with a blue whale, it's pretty clear you've got one individual. So animals really are spectacular. Sometimes when I sit around and imagine, like, if I was a fern, you ever do that? Sit around and think, if I was a fern, I'd like to think, even if I was a fern, I'd think animals are really cool. It's easy to think animals are cool because we are animals, right? So I'm obviously very animal-centric and I'm biased, but I'd like to think even if I was a fern, I'd probably still think animals were pretty cool. Animals are really a remarkable group of organisms. It's in many different ways, both from anatomical complexity to ecology to behavior, which we used to argue about whether non-animals have behavior. Definitely different than what you have in animals. Animals just are a really interesting, spectacular group, and it's a group that's been recognized for a very long time. Now remember that historically the group that we call animals would have included things like protozoans and stuff that have now been pulled out. But when Whittaker sort of came up with his five-kingdom system and made the delimitations of the different kingdoms a little bit better than what they had been before, that group that he recognized as being animals is largely what we would recognize as being animals today. So animals, even has historically defined, if you go back maybe as far as Whittaker, is a monophyletic group. As long as you don't have things like protozoans and stuff in here, animals really are considered to be a monophyletic group, and there's lots of evidence to support that they are. So what makes an animal an animal? Animals are all eukaryotic. Yeah, there's no exception to that. They're all heterotrophic, with the exception, of course, you see, I say, well, all animals are heterotrophic, and I would have said that categorically a few years ago, and then, of course, a couple of years back, somebody brought me this paper talking about a little sea slug that has these chloroplasts that are inside of its cells. They've actually transferred part of their genome to the host cell, to the host nucleus, right? And they're passed vertically from one sea slug to another, right? So, like from parent to offspring. Yeah, that starts to really sound like a photosynthetic animal, right? I'd be hard-pressed to look at that objectively and not sort of admit that those are photosynthetic animals. That's very different from Cassiopeia that you saw in lab that has the zooxanthellae that live in tentacles. Those are not intracellular. They're not living inside of the Cassiopeia cells, right? So there's a close symbiotic relationship, but it's not an intracellular relationship. And there's lots of intracellular parasites and things. You wouldn't consider those to be organelles either. But once you have them starting to pass major portions of their genome to the host nucleus, then that's when we start to think of them as organelles rather than as just endosymbionts, right? So somebody convinced me, and there's a paper, if you look under the lab supplements in Angel, that talks about this little photosynthetic sea slug. I was very skeptical initially, but the more that I read it, if the way that these researchers have described it is correct, it sounds like a photosynthetic animal. But most animals are heterotrophic. This definitely, To distinguish them from the type of heterotrophism that you see in fungi, most animals ingest food. So they bring food inside of a cavity in their body and digest it internally. Thank goodness, right? Because it'd be kind of gross if we're all digesting everything on the outside. But we don't. We bring the food inside and digest it inside. That's like a, sort of a hallmark of animals. All animals are multicellular. This is the way that Whitaker pulled out things like amoebas and paramecia and argued that they're not animals because they're unicellular. We realize now that they're not even related to animals, right? So they clearly don't belong in there. You'll see that some things like the choanoflagellates that are closely related to animals, you could make an argument that they're unicellular animals if you wanted to do that, right? And I'll show you at some point you could even make an argument that sponges are not animals, right? But most people don't consider choanoflagellates animals, and most people do consider sponges animals, but it's all kind of a matter of where you want to draw that line on the tree, right? Okay, this I challenge you. All animals have an animal life history. Go out there and find an exception to this. None of you is going to do it now, but I do not think there is any exception to this at all. All animals do meiosis to make gametes. Those gametes are the only haploid cells anywhere in the life history. They do not do mitosis. There is no haploid mitosis in any animal. Those gametes fuse to make a diploid zygote that might go through different juvenile stages or embryonic stages, but ultimately becomes an adult that produces gametes by meiosis. I'm not going to be asking you questions about animal life histories because they're boring. Animal life histories, animal sex is very boring, right? There is only one adult. There is no such thing as an alternation of generations. Even books that show you something like Obelias having an alternation of generations going back and forth between a polyp and a medusa and a polyp and a medusa, the polyp is the juvenile form, right? It becomes the medusae, right? So there's no alternation of nothing there, right? There's still only one adult, right? So you don't consider the polyp form to be a separate adult. It's not. It's a juvenile form, right? They all have this animal life history. You might remember fungi have a special way that they synthesize lysine, which is one of the amino acids. Animals have lost the ability to do it. So you get lysine through your food. It's one of the 8 essential amino acids. You don't get lysine, you die, right? If you don't get it in your food, you can't make it at all. If you're looking for a synapomorphy, something to put on the branch leading the last common ancestor of all animals, muscles, nerves, that'd be a really good one. There is nothing like muscles or nerves outside of animals. These are uniquely animal tissues, right? Muscles are obviously cells that are capable of contraction, and nerves are cells that are specialized to carry electrical impulse. I think I mentioned to some of you, a lot of people say, if you pluck a leaf off the tree, does it feel it? And I always answer that, I'm not presumptuous enough to know what a tree feels, so I'm not going to really try to answer that. But at the same time, if it does feel, it's got to be feeling in a very different way mechanistically than the way that we would feel something. It has no nerves, right? So it's not sensing you plucking the leaf off through some kind of nervous impulse, right? Because it doesn't have it. It doesn't have the same type of machinery or the same type of tissues that we have that would allow us to feel. And they also don't have cells that allow for like contraction or rapid movement. Going back to my high school kids, when I would ask them what's a characteristic of all life, they'd all start out by saying living things move, right? Which of course is not a characteristic of life, because there's lots of things that move that are not alive, like clouds and the sky and waterfalls and stuff, right? So movement is not a characteristic of life, but the reason that they would say that is because they think of an animal. When you say a living thing, nobody thinks of a mushroom or a tree, they think of a panda bear, or something like that, and of course they picture it moving, right? And so to them, a characteristic of life must be movement. But you can say that kind of for animals. One of the characteristics for animals is that most of them are capable of movement, and usually rather sophisticated movement compared to non-animals, right? And that's largely due to the fact that they have these muscles and nerves. Have you all heard of Hox genes? Hox genes are a family of genes that determine anterior-posterior patterning in animals. So it's important when we're going from one little cell, which is the zygote, and dividing in order to make the trillion or so cells that's you, that we get your head on this end and your tail on this end, right? Otherwise you're going to be kind of funny looking. So there's actually a series of genes, a family of genes that control laying out your body so that it gets laid out correctly in the right order, anterior to posterior. These are unique to animals. All animals have Hox genes, and non-animals don't. So again, if you're looking for a synapomorphy, something that would define animals as a group, Hox genes would be good. Fungi don't have Hox genes. Plants don't have Hox genes, right? They don't have heads and tails, if you think about it, right? So they kind of, it would make sense that they don't have
anterior, posterior, what?
patterning or development, right?
Development.
They determine which end is going to be which, kind of. Animals don't have cell walls. You probably knew that. And that might seem, oh, well, that's no big deal. That's really weird, right? Most other organismal groups have cell walls, with the exception of some of the protista groups. I mean, all plants have cell walls. All fungi, again, somebody found me a weird example of a little fungus without a cell wall, but all fungi have cell walls for the most part. All the different algae have cell walls. Virtually all the bacteria, except for intracellular parasites, have cell walls. Because remember, a cell wall allows you to osmoregulate for free, right? Basically sets up hydrostatic pressure around your little cell membrane so that it doesn't explode as fresh water comes in. Animals, because they don't have a cell wall, have to, most of them, osmoconform in one way or another, which is energetically expensive, but that's what animals do. So this idea that they don't have a cell wall, that's a really interesting trait. You could sort of put that as a characteristic that defines animals. It's unusual not to have a cell wall. And then animals have these, we looked at other things that have extracellular matrices as well, but animals tend to really have lots of elaborate extracellular matrices, often with collagen and glycoproteins. Sometimes they're calcified to form like bone or shell or spicules. And these are really important skeletal elements that make up the skeletons of animals. If you think about it, if you have cell walls, it's almost like you have a little skeleton around every cell in your body, right? But if you don't have cell walls, animals tend to have skeletal systems that are sort of separate from a majority of the cells in their body, right? So that they have sort of these skeletal systems, right, that are separate from the majority of the cells. Yeah, so could you all define an animal? What makes an animal an animal? Okay, and I've done all this already, right? So I'm going to go through it very quickly. Before, when we talked about fungi and plants, we didn't focus on things like body symmetry, because most of them are asymmetrical, even though like plants can have portions of their body, like flowers or something that are, or leaves that are symmetrical in one way or another. But in animals, it becomes important, so we talk about it a lot. There's some animals, like sponges, that are asymmetrical, that really don't have body symmetry. But most of them can be categorized as being either radially symmetrical or bilaterally symmetrical. So if you're radially symmetrical, it basically means you're round. There are multiple different planes that you could pass through a central axis that would give you two mirror image halves. If you're bilaterally symmetrical, it means that you have a left and a right? It means that basically there's a single plane that you could pass through the animal and end up with two mirror image halves. So you're bilaterally symmetrical. Most of the animals we're going to look at are bilaterally symmetrical. Yeah. You'll notice an interesting thing I can kind of point out here. You'll notice that if you're bilaterally symmetrical, you can talk about an anterior and a posterior end, right? We tend not to use those words for something like a sea anemone that's radially symmetrical. Instead, we talk about the oral side and the aboral side. So the oral side is the side that the mouth is on, right? And the aboral side is the side that's kind of like abaxial, means it's what? Facing away from the axis, right? So the aboral side is the side facing away from the mouth, right? Okay. I mentioned this briefly, right? All animals start with a zygote, and then that zygote starts to divide 2 cells, 4 cells, 8 cells, 16 cells, 32 cells, right? And during development, that animal becomes initially a little hollow ball of cells called a blastula. And the blastula is fluid-filled. It has a fluid-filled cavity called a blastocele. This is very distinctive of animal development. You don't find this outside of animals. And virtually all animals go through a stage that looks like this. I think I mentioned to you in fossils, they actually look for little fossilized blastulas. That's considered to be evidence of an animal, right? Because there is, I mean, we've already studied all the other groups. Do you remember in the fungi me showing you something that looks like this? Or in plants, did I show you something that looks like this? No, there's nothing, right? I mean, this is something you haven't seen before, and yet it's developmentally characteristic of all animals. For most animals, remember, they're going to ingest food. They're going to have a primary body cavity that they're going to bring food into in order to carry out digestion. And that primary body cavity, or that GI tract, digestive tract, gut, whatever you're going to call it, initiates through an invagination into the blastula, right? Called gastrulation is this process. And what it does, not only does it set up an internal cavity that's going to become the gut, that's called the archintron, it also sets up 2 different germ layers, or tissue layers. It sets up an inside tissue layer, which is called endoderm, and then an outside tissue layer that's called ectoderm. So if you were to look at like the lining of your digestive tract, that's all endodermally derived tissues, right? Ectoderm would become things like skin, stuff that's on the outside. And for most animals, they actually have a tissue layer that develops in between the endoderm and the ectoderm, which is called mesoderm. Yeah. So if you have only two different tissue types, if you have only ectoderm and endoderm, we say that you are diploblastic. So for example, the cnidarians are considered typically diploblastic. They have only endoderm and ectoderm, no mesoderm. Although that's somewhat controversial, I'll tell you when we get to cnidarians. But that's typically the way that you're going to see it, like in a textbook. If you look at animals like us, for example, we do have that third layer. We have endoderm and ectoderm, but we also have mesoderm. So we're not diploblastic, we're triploblastic, right? Triploblastic just means you have mesodermal tissue, right? In addition to endoderm and ectoderm. Yeah. Okay, so you got to get really good with these words like, and I can tell you most animals that have mesoderm, the mesoderm actually fills in that blastocele. So the blastocele disappears over time. There's a group of animals, though, that actually retain that blastocele in the form of a pseudocelom, but we'll get to that in a second. And you see there's a ton of vocabulary here, right? Are you all getting good with blastula, blastocele, archenteron, gastrula? You'll also notice that there's a little opening here going to the archenteron. That's like an opening going to the gut, right? And so that opening is called a blastopore. And what's that going to become? What would the opening to the digestive tract be? It's going to be either the mouth or the anus, and it's going to depend on what kind of animal you have as to what that becomes, right? So, yeah, you'll see that as we go along, right? But remember on those, in the lab manual, it asks you fate of the blastopore. That's what it's basically asking. It's asking you, does it become the mouth or does it become the anus, right? Okay, this shows you an actual blastula undergoing gastrulation. So this is going to be the blastopore right here, and this is going to be the archenteron there where it invaginates. I can kind of see where the archenteron is right here through all the cells. So that shows you what it really looks like, right? Okay, as far as digestive tracts, there are two primary types. In one type of digestive tract, there's only a single opening, right? So in the case of something like this little polyp, if this was a sea anemone, there's just one opening right here. Food comes in, waste goes out, it eats and defecates through the same hole, right? So there is no one-way digestive tract. We tend to say that this is blind or that it's closed, right? That there's only a single opening. This is your little friend, Dugetsia. Notice he also has a closed digestive tract. This was interpreted for a very long time because this is the only opening to it right here at the end of the pharynx there. This was interpreted for a long time as being primitive, right? But if you look, this is his GI tract. See how elaborately branched it is? He has one anterior branch, two posterior branches, and then all of these, you were supposed to have looked at this on prepared slides as you look at it. All of these highly branched little diverticuli that carry, it basically, he doesn't really have a circulatory system, so that digestive tract is taking food to all different little parts of his body so that just through simple diffusion it can pass to every cell in his body. It's a really elaborate digestive tract, and yet this was touted as being primitive for a long time because it only has a single opening. Of course, we now know that these guys probably are closely related to other animals that have an open digestive tract, and that this is probably a reduction, right? In the case of the tapeworms, which are in the same phylum with this, guess what kind of a gut they have? They typically don't have a gut at all. So that means they don't ingest food. Why don't they ingest food? Where does the tapeworm live? It lives floating in somebody's intestine, right? Bathed in digested food. Why is it going to ingest that? It's just going to absorb it through its skin. The food's already digested. It's floating in digested food, right? But it's not without a digestive tract because it's primitive. That's a highly derived adaptation to the very specialized type of parasitic lifestyle that it lives, right? So you've got to be very careful when you look at something that looks simple to interpret it correctly, right? A simplification is not always the ancestral condition, right? In fact, it's not going to be if it's a simplification. If it's a simplification, it's a derived condition, right?
I was wondering, the worm that I brought in that we found on that, out the green alga stuff, where does that guy lie at?
it's a nematode, if I'm remembering correctly. The little one that went back and forth side to side, it's
Yeah,
a round
it's
one.
like chilling with this Spirogyra.
I'm going to show it to you in a second. Yeah, it's a nematode.
Nematode.
Yeah. So these are flatworms, those are roundworms.
Oh,
It's a common name for them. This shows you an open digestive tract, so obviously like yours is open. Most animals are going to have an open one. This means that there's a mouth and an anus, and food moves through in just one direction. Yeah. Okay, so for the animals that have an open digestive tract, which is clearly the one you have to be talking about here, remember we're going to say that if the blastopore becomes the mouth, we call that group of animals protostomes, which means mouth first. And if the blastopore becomes the anus, we say that they are deuterostomes, which means mouth comes second, and then the anus is the blastopore, right? The blastopore becomes the anus, and the mouth develops secondarily. You'll notice, too, that these animals, they're now showing this red tissue layer here. That's going to be the mesoderm. So you have endoderm, ectoderm, and mesoderm. So examples of protostome animals, things like mollusks, like snails, clams, squid, and annelids, things like earthworms and leeches. Those guys are protostomes. So their blastopore becomes their mouth. Deuterostome animals would be things like echinoderms. Those would be like starfish, sea urchins, right? And chordates, which is going to include all of the vertebrate animals, including us, right? Those would all be deuterostomes, so the blastopore becomes the anus, and then the mouth develops secondarily. Okay, so now we come to the coelom type. So this is a platyhelminth, right? This is a nematode here. And this is an annelid, a segmented worm. So flatworms, roundworms, segmented worms. Hopefully by now you realize the word worm means nothing. It means you're long and skinny. There's no close relationship between any of these. This, for example, right, if I was to ask you, this right here is more closely related to like a grasshopper, believe it or not. And this one is more closely related to a snail. So these are not related to each other at all, right? Only very distantly are they related to one another. So just because something's a worm doesn't mean it's related to other things that are worms, right? This is typically used to show this anatomical idea called a coelom, right? And I said a lot of people have trouble understanding this for some reason. I've never quite understood why, but I'll try to help you. So anytime you see C-O-E-L, that refers to a cavity. And this, a coelom, when we talk about it in animals, is a secondary body cavity, right? So the primary body cavity in an animal is the... What's the primary body cavity that we just talked about forming? About the gut, right? Or the digestive tract. That's the primary cavity. The primary cavity, the first cavity that forms in your body, is the cavity that you're going to put food into to digest it. So that's the primary cavity. But you have other cavities in your body besides that, right? Including one major one. So if I asked you, for example, where are your lungs? Where's your liver? I mean, they're inside, right? They're not hanging off your arm out here. And inside where? They're not inside your stomach. They're not inside your intestines. Where are your internal organs? They're inside your coelom. That's what a coelom is, right? In the case of mammals, the coelom is divided by a diaphragm so that you have an abdominal and thoracic cavity, right? But those cavities together make up your coelom. So there are some animals that don't have a coelom at all. All the animals you looked at in lab this week don't have a coelom. So if you looked at this little flatworm here, you'll notice he has a primary body cavity. He has a gut, right? But after that, you have endoderm, mesoderm, ectoderm out here. There's no other cavity in his body. There is no other space. He doesn't have complex internal organs that occupy some kind of a cavity, a larger cavity inside of his body, right? He really only has a primary body cavity. There is no secondary body cavity. Nigerians and platyhelminths are not going to have this. This was the other reason that people argued that these were primitive somehow, is because they're acoelomate is the way that we say this, right? A just means without. They don't have a coelom.
Professor Bro?
Yeah.
I have a question about this primitive word that always comes up with all of these different animals, from like bacteria to fungi. Like, do they just think like everything is primitive, or like what?
Typically, people would interpret simple as primitive. Primitive, of course, is a word I told you I didn't even want you to use, right?
Yeah, everything it seems.
I'd rather you use the word ancestral. The trait, because primitive sort of implies that if you're primitive, that means you're the ancestor, right? Is what that word kind of implies.
If you say that you exhibit ancestral traits, then that just sort of means you look like the ancestor, not that you are the ancestor, which is obviously ridiculous, right? Because these are all modern organisms that are in the world today. None of them are primitive in the sense that none of them are ancestors. They're all modern, right? But at the same time, when people would talk about an organism being primitive, what they're talking about is that they think that it's exhibiting ancestral traits, that it looks a lot like the ancestor of a larger group would have looked, right? The problem is the traits that are interpreted as being ancestral are usually simple traits, right? And simple does not mean necessarily ancestral. Sometimes simplifications, many times, simplifications are actually a derived condition. It's actually something new. It's not something old, right? So snakes, for example, don't have limbs. That's not because they're exhibiting an ancestral trait, right? They've lost limbs on purpose
So it'd be
because
something
of the
else
type
new,
of
right? That'd be
locomotion.
derived.
Huh?
It'd be new and derived, right?
It'd be new is derived, old is ancestral, right? And so if you say something's primitive, what you're saying basically is that it's exhibiting the old traits, right? We would say that bacteria exhibit a lot of ancestral traits because they're presumed to have a lot of the simplicity that they have represents sort of what the
Yeah, I was
ancestral
saying
form
the
of
loss
life
of the
would look
snake's
like.
legs, that would be derived, new, right?
Well, right, well, that's the thing. There's this misconception that somehow to be new or to be derived has to mean that you're increasing complexity. That's not true, right? This is one of the misconceptions that people have about evolution, is that evolution is constantly increasing complexity. That's not true. There's zillions of examples where you reduce complexity or you streamline something, and that's, as long as selection favors it, that's the way it's going to go, right? So, for example, yeast are unicellular, right? They're not filamentic. That's a derived condition from them, right? They're exhibiting sort of an ancestral trait, but the unicellularity that you see in them is actually derived. It's not ancestral, right? Because they're evolved from filamentous ascomycetes. There's lots of examples of this where a reduction in form or a simplification of form, and these guys are probably a very good example of that. They probably lost an anus at some point rather than that they don't have an anus because they're exhibiting an ancestral trait. It's probably a derived trait for them. Yeah. You just have to not think that simple always means ancestral, right? Because it doesn't many times.
What about a skink? Have you seen one of those?
Yes, what about a skink? Skink's like a lizard.
Yeah, it's like it's snake with wings.
Well, it would be probably a lizard, though it's a squamate.
Yeah.
So lizards and
Okay.
snakes are each other's closest relatives
Yeah.
within modern reptiles. But snakes, as far as I know, are monophyletic groups, right? So they're a derived group. I don't know that lizards are. I would kind of, that's
It's
an
a
interesting
snaky
question.
little thing.
Right, and notice that a lot of lizards sort of have, they operate longitudinal muscles on opposite sides of their body, so they have something almost serpentine but with legs. That would be a really good thing for somebody to look up, because of course I don't study that group at all. My guess would be that lizards are paraphyletic with respect to snakes, and that snakes are a monophyletic group, but I don't know. Lizards and snakes might be reciprocally monophyletic within squamates. Somebody's going to look that up. Look that up and tell me whether or not lizards are a monophyletic group. I would guess that snakes are and lizards aren't, because snakes can be defined by a derived condition, a synapomorphy, right? The loss of legs. But I have a feeling certain lizards are more closely related to snakes than they are to other lizards, but I'm not sure that's true. Okay, so acoelomate means there is no secondary body cavity. Pseudocoelomate means you have a secondary body cavity, and the example that's always used for this are nematodes, but there's other pseudocoelomate animals, things like rotifers, for example, are pseudocoelomate too. They have a secondary body cavity here that houses some internal organs, in addition to the primary body cavity, so primary body cavity and a secondary one. But you'll notice that this secondary body cavity is not completely lined with mesodermal tissue. It's not completely touching mesoderm everywhere. And it also has a very different origin. You'll see that the origin of this cavity is actually the blastocele. So remember, in most animals, the blastocele fills in completely with mesoderm. In pseudocoelomate animals, it fails to fill in completely with mesoderm, such that you're left with this false coelom. A true coelom has to be completely lined with a peritoneum, with a mesodermal layer, completely touching the cavity everywhere. And this one, you can see you've got endodermally derived tissue touching that cavity. So by definition, it's not a true coelom. Yeah. This was considered to be the most primitive. This is a little bit better, right? And this was considered to be the best. Ends up not being the way you should think about it, but that's the way it was interpreted for a very long time. True coelomate animals, something like an earthworm, you'll notice that they do have a primary
cavity, obviously, the gut, right? And then they have a secondary body cavity that houses internal organs. This qualifies as a true coelom because it's completely mesodermally lined. I'm gonna, in the lab, like in the next couple labs, you're gonna see cross-sections, prepared cross-sections of earthworms and nematodes. I very often put that in the practical. And I just ask you, does this animal have a pseudocoelom or a true coelom? It's very easy to tell once you've seen them, right? Because in a nematode, you'll see that you do not have mesodermal tissue touching every part of the coelomic cavity, but you do in an earthworm. So if you have an animal with a true coelom, you can talk about how that coelom arises. What's the origin of that coelom? Remember, a pseudocoel is derived from the blastocoel, right? But if you have a true coelom, there's two different ways you can get it.
What, did you say what exactly the function of the mesoderm was?
It gives rise to a bunch of tissues that, like most of your internal organs and things, are mesodermally derived. Most muscle, I think, is mesodermally derived.
So it's like precursors to muscle?
Like skeletal muscle and stuff, I'm pretty sure is mesodermally derived. It can be. It depends on the animal, right? Like, for example, Nigerians have muscles for sure, and they don't have true mesoderm, right? So there are other ways to get muscles, but like in you, most of your skeletal muscles and stuff, I'm pretty sure are mesodermally derived, right? Whereas you have external tissues and organs that are ectodermally derived, and then internal things that are endodermally derived. It just is sort of, it refers back to these three discrete layers that you can see in very early embryonic development, right? And each one of those basic germ layers gives rise to a certain set of tissues that are either external, in the middle, or internal, yeah? Would you ask me what's the advantage of having it? I would imagine it allows for more anatomical complexity. If you have only two germ layers, you tend to be anatomically simple by comparison. How's that for an answer from somebody who studied mushroom systematics? I don't know, really. That would make sense. Okay, so here you see, obviously, a triploblastic animal that has ectoderm, endoderm, and mesoderm. This would be an animal, most protostomes that have a true coelom do it like this. They form what's called a schistocele, right? So schizo or schizo means split, right? This comes from a splitting of mesodermal tissue, and like this is going to be the coelom. So it arises through this splitting of mesodermal tissue. In the case of an intracoelous coelomate animal, something like you, or we're going to look at starfish, right, in the lab, and you'll see that here the coelomic cavity actually develops from outpocketings of the arc intron. So it's the arc intron that actually provides the original cavity that gives rise to the coelom. Yeah. In the case of a pseudocoel, though, which isn't a true coelom, it arises from the blastocoel. So splitting of mesoderm versus outpocketings of the arc intron versus blastocoel, right? Those are the three sort of different ways that you can end up with a secondary body cavity, two of which are considered a true coelom and one of which is not. Okay, and then we come to this sort of bad thing of cleavage. And this refers to, we start with a zygote. When the zygote divides the first time, you end up with 2 cells, right? And then you're going to go 4 cells, 8 cells, right? So in an animal that has determinate cleavage, what happens is when that zygote divides the first time, those 2 cells, at least to a certain extent, those cell fates are already locked in. They can't just go ahead and start doing any old thing. The cells have already started to differentiate to the point where maybe one is going to be a head, it's going to give rise to all the stuff in the head, and the other one's going to be a tail and give rise to all the stuff in the tail. And if you pull the 2 cells apart, they can't develop into a complete individual because they've already been fixed into developing into only a certain part of them. So an animal that has determinate cleavage, some type of determinate cleavage, the cell fates are established really on the first division of the zygote. If an animal has indeterminate cleavage, what this means is you could go through a couple rounds of mitosis, pull one of those cells away from the little cluster of 4, and each one can develop now into a complete individual. They could just start over again. They could start dividing again. So the cell fates are not completely locked in right on the first division of the zygote. Obviously, which kind are we, do you know?
Indeterminate.
We're indeterminate. How do you know that?
Breeders.
Huh? Because we can have identical twins, right? Something that has determinate cleavage is incapable of producing identical twins, right? Because the moment the zygote divides, the cell fates are already set.
Now, I've had students come to me and get confused with this before because they said, you mean our cells never differentiate completely? They never are determinate completely? And of course they are, right? I can't just like go pluck one of your skin cells off and like grow a new you, right? And that's not going to work. You obviously have highly differentiated cells that are completely determined relatively early on. But what we're talking about here is very early on the very first divisions of the zygote, whether or not the cell fates are locked in immediately or whether you can go a little bit, a few rounds of mitosis at least, before the cell fates are determined. This is probably the ancestral condition for all animals, to be indeterminate. And determinate cleavage in the old Campbell would have been described as being spiraled, right? Because if you look at this 8-celled stage, supposedly it looks like it's twisted. See how these 4 cells sit twisted on top as opposed to these sit directly over them. Yeah? This is impossible to see. I think I mentioned to some of you, at San Francisco State University, in one of the labs, they used to use sea urchin, which are deuterostomes, right? They have supposedly radial cleavage like this, and crepidula snail slides, which have supposedly spiral cleavage. And the students were supposed to tell these apart. I can't tell them apart. There's plenty on the sea urchin slide that look exactly like this, and there's a whole bunch on the crepidula slide that look a whole bunch like this. I can't tell them apart. But I don't feel so bad, because even the people that study this, they're now starting to acknowledge that there's actually a lot of different types of determinate cleavage. Determinate cleavage patterns have probably evolved a dozen different times across animals. Yeah. So all determinate cleavage is not equal to all other determinate cleavage. And in fact, Campbell now acknowledges at least a second type of determinate cleavage that they call I. So this is R, this is S. They call it I. I stands for idiosyncratic, which means weird. They don't even know how to describe it, so they just call it weird cleavage. So we've got radial, we've got spiral, and we've got weird. And those are the 3 that they acknowledge. There's a good paper in the lab supplements, again, on different types of determinate cleavage patterns in animals, if you want to look at it. I particularly don't like this term, because inevitably you're going to do what on the next exam that's going to make me drink more? You're going to confuse radial cleavage with radial symmetry. There's no relationship there, you realize. One is talking about overall body symmetry. The other one is an unfortunate name that you're going to see over and over again to refer to indeterminate cleavage. So if you talk about radial cleavage, what that means is that the cell fates are not established early on in development. That's what that means. It has nothing to do with being able to divide it and get 2 Merriam-Websters or anything, right? They're 2 totally different things. So can you please make my life better and not confuse radial symmetry with radial cleavage? For the most part, I'm going to try to refer to this as indeterminate cleavage, but you're going to see that radial a lot, yeah? I keep hitting that thing wrong. This shows that this is out of Brooker, right? So here it shows if you have sort of a protostome animal, that's what they're saying, the blastospore becomes the mouth. Here's the 4-celled stage. If you pull one cell away, what happens? Nothing, right? But here, if you have a deuterostome where the blastospore is becoming the anus, typically they have this indeterminate cleavage. You pull one cell apart, everything can become a normal embryo, right? You can end up with 2 identical twins. So they're going to argue that mollusks, platyhelminths, and annelids have spiral determinate cleavage, that arthropods and aceals have idiosyncratic. I'm going to argue that aceals, you'll see where they fall phylogenetically. This has to be something different than the one that these guys have. But I'm going to argue that arthropods, nematodes, onychophorans, and tardigrades have this type of determinate cleavage, whatever we're going to call this weird cleavage, right? And then the rest of everybody has radial, which kind of is, and that's not really even true, because remember we said ctenophores have some type of determinate cleavage, again, independently derived from these, right? But all of this smattering of organisms, of animals that have radial cleavage, presumably that's the ancestral condition, right? And all of these different determinate cleavage types are derived. Most animals have some kind of a skeletal structure. You need something rigid to work muscles against. You have an endoskeleton, because it's on the inside. If you look at something like a sea urchin test, it's also an endoskeleton. There's a skin that goes over the outside of it. Arthropods have an exoskeleton. It's on the outside. They're squishy and soft on the inside, but they have a hard outside skeleton. And then something even like an earthworm has to obviously have something akin to a skeleton so that it can move. It needs to have something rigid to work muscles against. And so here you see This is its coelom, right? This is the GI tract right here. This is the coelom. This is actually mesodermal tissue. These are longitudinal muscles, which are mesodermal tissue, right? So you've got mesodermal tissue completely touching this. It's a true coelom that arises as a schizocele. That coelom is filled with fluid. It's a non-compressible fluid, and it can be made rigid so that the muscles work against that. So these longitudinal and circular muscles are basically working against the rigid, fluid-filled coelom.
So if it was a pseudo-sloam, there would be no white stuff in there?
There would be no mesodermal tissue here.
Is that,
You'd have only, this is endodermal tissue here, and you'll see on the nematode that this endoderm would directly touch that cavity. You also see that where you have circular muscles here, you have no circular muscles in the nematode. They have only longitudinal. It's pretty easy to tell them apart. You're going to look at both of them in cross-section like that. You'll be able to tell them apart. But I almost always put that in the practical. So this is called a hydrostatic skeleton, right, where you're basically using a non-compressible fluid-filled cavity as a rigid structure to work muscles against. And then I already went through this whole thing with muscles, right? You all know that muscles never
Pull.
no, they only
Oh, sorry.
pull, they never push, right? Because muscles, the only force that a muscle can exert is in contraction. There's no force in relaxing, right? So muscles typically work in antagonistic pairs, right? If you have a jointed skeleton, one muscle pulls to bend the joint, and one muscle pulls to straighten the joint. Yeah? If you have sort of a long skinny body like an earthworm, or even you can think of your little hydra, the muscles are typically arranged, the antagonistic muscles are arranged as circular and longitudinal. When longitudinal contract, the circular relax, and you get short, fat. When the circular contract and the longitudinal relax, you get long and skinny. Yeah? Any animal you see that can scrunch, the way your hydra was scrunching, where he gets short and fat, remember the hydra gets short and fat? Then you let him sit there for a while and he comes out long like this. He's got to have those two sets, right? Otherwise he would not be able to go back and forth like that. If you had only one set, if they had circular muscles, this guy would go and contract the circular muscles, and now he's stuck. Yeah, there's no way to undo that. He can relax all he wants, and that's not going to come undone, right? He needs to have muscles that are this way that are going to contract in order to make him do this. Yeah. So if you have an animal that has a jointed skeleton, not just this is typical of vertebrates, but you have some invertebrates like arthropods, right, that have jointed skeletons. They have little flexors and extensors, kind of like we do, right, that bend joints. So there are variations on this. For example, in the squid, we'll look and see that they actually have a group of muscles called radial muscles, kind of like in a radial tire that go across the mantle. But most of the animals you look at, you're going to kind of be able to think of antagonistic pairs as being either circular and longitudinal muscles or being flexors and extensors. Okay, so let's try a couple of these. So the early gut is called the blastula, gastrula, coelom, or archenteron. What do we call the early gut in development? Okay, one more. There we go. Good Lord. So the archenteron, right? The blastula is the hollow ball of cells. The gastrula is the blastula after it's undergone gastrulation, after it has an archenteron, right? The coelom is much later on, right? That's a true secondary body cavity that houses internal organs. The early gut is called the archenteron, right? Why did so many of you miss that? No? Okay, diploblastic animals lack what? Endoderm, ectoderm, mesoderm, or true tissue?